All right, it's time to begin. This is lecture number 12. Topic is on blood. Yeah, I'm just making sure it's 12. Today's study tip says take a day off from work if you are stressed out for time, okay? This pays off sometimes, especially if you're working full time. If you're stressed for time to study for the exam, consider asking your employer for a day off. Some of you I know are kind of sneaking out to come to school. Your employers don't know. Some of you have a good relationship and they're happy to let you study because they know what it means to go to school. So hopefully you're able to give yourself, especially the day before would be really great for you to be able to focus and get some extra time to study for the exam. So this is chapter 13 on the blood. It's a fun topic. We all have blood, and of course it's a big part of nursing, a lot of you going into that area. We'll do a big lab on this to really help emphasize all the things we're going to do in lecture. And a lot of you have heard about these topics before, but hopefully you'll really nail it this time and get it all the way around with blood typing and where cells are made and all that kind of stuff. Okay? So there's a lot of information, so let's get started. If you take a sample of someone's blood and the name of a person, what's the name of the technician who does this job? What's the name of the person who takes the blood sample? They're called a phlebotomist. Yeah, phlebotomist is a certificated position. It's a good job if you're looking for something to do while you're going to nursing school or going to college. A phlebotomist, works with Unilab or other places like that. You sit down, they put on the tourniquet, clean the area, get a big needle. Grosses me out, personally. I can't look. You guys probably like that stuff. And then the sample is put into a test tube and they say goodbye. Well, if you take this sample and you centrifuge it, and a centrifuge, if you don't know, is a large machine. It could be a small machine, but it takes a large test tube, many test tubes, and spins them really fast. And we have a type of centrifuge that we're going to use in class called a microcentrifuge. And we're going to be doing the same thing. We're going to take some of your blood and we're going to spin it. And the heavier things fall to the bottom. Does that make sense? If you centrifuge, right? And the lighter things stay at the top. So in this case, the heavier things are called the formed elements, and the amount of formed elements from the bottom to this white line here is called the hematocrit. The top part is just called the plasma, and the area between is called the buffy coat. Usually in lab we can't see that very well, but we have very small samples, so perhaps in a larger sample you could see it. In the buffy coat, you would expect you could see some of these things over here in the blood smear, platelets, white blood cells, red blood cells. So it's kind of a mixture of both things. The top and the plasma is mostly white cells and water, proteins. Bottom, well, it's obviously red, so those are the red blood cells. Okay, the area between is going to have kind of a mixture of everything. All right, so let's focus on the lighter stuff, things at the top of the test tube. And that's called the plasma. The plasma is essentially the liquid minus the hematocrit. So whatever is not in the bottom hematocrit layer is what we call the plasma. It's 90% water. It has food, salts, oxygen, waste products, and proteins. And 3 examples of proteins to tell you about are albumin, which we have heard about before. Globulins. Globulins come from the word immunoglobulins, and immuno means your immune system. So these are antibodies. Immunoglobulins are the fancy name for antibodies. And fibrinogen. Fibrinogen is a protein involved in blood clotting, of making a fibrin. So we will refer to that again today when we talk about blood clotting. You may have also heard of the term serum. It's not the same thing as plasma. It's plasma minus the clotting factors. And the clotting factors are the things like fibrinogen and fibrin. So if you take someone's plasma, if you centrifuge, take the plasma, it could clot and therefore couldn't be used to do experiments because it's all gooey. So a lot of times they'll remove the clotting factors so they can look at the serum and have it remain as a liquid and not get all gooped up with clotting factors. Alright, so the formed elements is the stuff at the bottom, and this is where you're going to see the different types of red blood cells. So the examples of the white blood cells are given in the first 1, 2, 3, 4, 5, 6 pictures. The neutrophil, the eosinophil, basophil, lymphocyte, monocyte, and platelets. Those are all white blood cells. Now you should know these words. These should be familiar to you from anatomy. If you don't know it and you took anatomy a long time ago, get busy on your anatomy book. Go find it, go buy one, and learn all this stuff again. The neutrophils are the most common. We'll talk about each of these in lab more. And you're going to do a white blood cell stain. You're going to be looking for some of these lymphocytes. The monocytes are big, so you should be able to see them. Some of them are multinucleated, like the neutrophil, the eosinophil. Platelets are technically not cells, as are erythrocytes, because they both don't have a nucleus. So a platelet is buds off of, it's like a little piece that sheds off of a larger cell called a megakaryocyte. And it makes these little platelets, and they're very important even though they don't have a nucleus and a boss, you know, in the sense of a nucleus and what to tell it what to do. It's involved in making a blood clot, which would, of course, help you from bleeding. The erythrocytes are the most common of all of these cells, and erythro means what? Red, right? Red cells, erythrocytes, and we'll talk about them more. What's the technical term for a white cell? They're called leukocytes because leuko means white, right? So let's start with the formed elements, being the number one cell found there are going to be the erythrocytes, the red blood cells. And as you saw their shape, they're kind of disc in shape. They kind of look like a candy, some kind of candy to me. There's no mitochondria. They used to have it when they were first made, and then that they want to become small so that they can move through capillaries in really tight places. So to become smaller, you have to dump a lot of the unimportant organelles, and in this case, it's unimportant to have mitochondria because they don't. Mitochondria, remember, makes ATP, and unlike a muscle cell that's constantly using ATP, a red blood cell doesn't really need it. There's also no nucleus, so this means that it doesn't really do anything with a command of a brain, essentially, is what the nucleus is. And so mostly there's just chemical reactions happening in the formed elements. No changes in the cell, no growth of the cell. It can't divide. Since it has no nucleus, there's no chance for mitosis. So therefore, once it's made, it has a very short lifespan of about 4 months. 120 days, 4 months, whichever one you like. And, of course, you know, the color is red because it contains what? Contains hemoglobin. Yeah, hemoglobin has iron in it, and when iron becomes oxidized, for example, if you take a piece of iron and you go to the coast, you take a nail, an iron nail, and you take it to the coast where it's salty air, it's going to increase the amount of oxidization because of the salt. And what color and what happens to the nail? The nail becomes rusty, right? And rust is what color? It's reddish, and so we call it rust color. It's the iron becoming oxidized, which means an oxygen is binding to the iron, causes that red color. And so that's exactly what happens in hemoglobin. So in this picture, I think we've seen it before when we were talking about protein structure. Hemoglobin has 4 subunits, has 2 purple ones down here, and 2 blue ones at the top, so that's 4 subunits. And the middle of each subunit is what is called the heme group. And I'll show you the next picture. This is what heme is. bunch of carbons, hydrogen, nitrogen. In the center of the 4 nitrogens is an iron, Fe. And when the iron becomes oxidized, it turns reddish. So if there is no oxygen, then you won't get that color on this iron. So if one hemoglobin holds how many irons? 1, 2, 3, 4 irons, and therefore how many oxygens are on one hemoglobin? Same, 4, right? There's one here, one here, one here, and one here. So this is how hemoglobin works to pick up the oxygen, is it has iron, and the iron jumps on and jumps off, and the oxygen jumps on and jumps off the iron. There are some problems like anemia, where someone has low iron in their diet. And if you have low iron in your diet, you might only have 1, 2, 3 irons. Even though the hemoglobin is complete, this one might not have an Fe in the middle. So you'd only be able to hold 1, 2, 3 oxygens. And of course, how would you feel? How would a person feel if they had a lack of iron? Sure, they'd feel slow, lethargic, they wouldn't have much energy, and you can see the anemia being the problem for that. There's different kinds of anemia we'll talk about in lab. That happens to be one called ferrous anemia. Ferrous means Fe, iron. Okay, the formed elements continue. The leukocytes are the white blood cells. If you look at them under the microscope, some of them have little dots that you can see, and so we call them granular leukocytes. The neutrophils, I told you, are the most common. So when you do the slide in class, you're going to see a lot of neutrophils as the number 1. And the eosinophils, the eosinophils are involved in your allergic reactions, and they are of course not going to be used all the time, so therefore there's not as many of them since you don't walk around having allergies all the time. Some of you more sensitive to allergies than others, you would therefore have more eosinophils. Basophils, also involved in skin in any kind of damage to the skin or muscle or tissue, also could be involved in allergies, and that's to make histamines. So you probably know the word antihistamine. Antihistamines you can buy are Claritin, Zyrtec, Allegra, all those, Benadryl. Those are antihistamines, and they prevent the histamines from working. So if you have a runny nose due to allergies, you take the antihistamine, and what happens to your runny nose? It stops, right? So the purpose of the histamine is to increase blood flow to an area, causing swelling, and it's actually trying to attack, or in your nose, it would be trying to attack some pollen maybe that you just were breathing. So histamines are a good thing in order to attack the foreign invader that's come into your nose, but it becomes annoying to blow your nose all day, so we have drug companies that have introduced products that inhibit the body from doing things, and so we now take antihistamines if our allergies become too severe. Okay? Agranulocytes. A means what? It means without, right, so these are without the granules. The monocytes are some of the largest leukocytes that you have, and since they're so big, they do a great job of doing phagocytosis. What's phago mean? We already learned this. To eat. Yeah, to eat other cells. The monocytes are the big eaters of a bacterium or other big things that come into your body. Lymphocytes are the T cells and the B cells. So they are part of, they are derived in your lymphatic system, so they are found in lymph nodes, so that's why they have the word lymphocyte. So we'll talk more in your immune system about all of these when we get to the immune chapter on T cells and B cells. The last one are the platelets. The technical term for the platelets are thrombocytes. And thrombocytes, platelets are involved, as I told you, in blood clotting. Okay? Alright, next topic. This is a big word. Can you say hematopoiesis? Hematopoiesis. Say hematopoiesis. And then you can also use the good. The word hemopoiesis. So they're both very similar. It's the point of it is the word hemato or hemo means what? Means blood. So hemato and hemo mean blood. And poiesis means the making of. Okay, so the making of blood cells is called hematopoiesis. And they all derive from cells called pluripotent stem cells. Pluri is like the word plural. What's that mean? That means many. And potent is like having the potential to become. So there's many potentials of these cells. They can become any of the cells that we just listed previously. Where do you find pluripotent stem cells? In the red bone marrow. And from anatomy, you'll remember children have a lot of red bone marrow in all these different places. But in adults, a lot of our red bone marrow has been placed with yellow bone marrow, and now we mostly have it in just our sternum, the ribs, and the hips. Okay, so more specifically, if we want to talk about making erythrocytes, we use the word erythropoiesis. So erythropoiesis is the making of the, I write RBC, red blood cell. Now on the test, you don't want to write RBC, you don't want to write red blood cell. What do you want to write? You want to write erythrocytes. That's right, you want to impress me that you know the technical terms, and that's what you're going to do, okay? The kidneys monitor the red blood cells, and that makes sense because 1 drop of blood flows through your body and your kidneys every minute. So your kidneys are really great at monitoring your blood because they see it so often every minute. And it can say, hey, whoa, problem here. The erythrocytes are low, and so we need to help the body increase them. So to do that, the kidneys release a hormone called erythropoietin. Try saying that. Try saying erythropoietin. Now, to make erythropoietin means that it's actually an endocrine gland. So remember we learned about some glands have mixed functions? This is one that has a mixed function. Obviously the number one job of the kidney is to produce urine, right, and clean the blood. But it's also an endocrine gland. It makes the hormone called erythropoietin. There is a disease, lots of diseases of course, but the one that I'm thinking of in this case is when there's too much erythropoiesis, too much erythropoietin, it causes polycythemia. Poly means many, many red blood cells. Polycythemia, too many red blood cells. If you increase the red blood cells, what happens to the viscosity, the thickness of your blood? If you add a bunch of red blood cells to your blood, what happens to the thickness of it or the viscosity of it after you added so many red blood cells? Does it become greater or lesser? It becomes greater, right? It becomes more viscous. Now you ask that fluid to go flowing through your body, and now it's really thick, like ketchup. Is it going to be able to pass easily through your body? No, it's not. It's not going to travel well. A person's not going to have delivery of oxygen to, especially like to the feet and the hands, because the heart is going to have a harder job pumping that really thick fluid. So it's very dangerous, very dangerous. Now this brings up a story of athletes who kind of would like to have extra red blood cells. Why would it be nice to have extra red blood cells? Well, red blood cells, hemoglobin, what do they carry? Oxygen. Right, they carry oxygen. And remember we were talking about aerobic conditioning. Supplying more oxygen to your tissues will mean that you can make more ATP and you won't have any lactic acid buildup. So they want more oxygen delivered to their tissues. So there is, what's it called? I can't think of the drug, but there is a drug that is similar to erythropoietin. I think it's just called poietin. And it would be given to somebody who might be having low red blood cells, and a doctor would prescribe it. But if an athlete gets a hold of it, a normal red blood cell count now takes the erythropoietin drug. They are now putting themselves at risk for having more viscous solution of blood. They could die from it. They also are, I mean, they're messing around with the wrong thing, right? But they're trying to enhance their performance. So this is one of the things they would test for in Olympic athletes. Are they taking erythropoietin to increase red blood cell count? There is another way that you could do this, and it's a correct way that your body would do this. If you move to Lake Tahoe or Denver, Colorado, where there's a lack of oxygen in the air because of the decreased pressure, and we'll talk about this more when we do respiratory system, but there's a lack of oxygen because there's a lack of pressure at the higher altitudes. If you moved to Denver or Lake Tahoe, immediately your body would go, whoa, we're not getting enough oxygen to our feet and hands and tissues. We need to make some more red blood cells, and it will. It will release erythropoietin, and it will make more red blood cells. This will take between 2 and 3 weeks for you to acclimate to the higher altitudes. So imagine an athlete. The race is in San Jose, where we're only 50 feet above sea level, decides to move to Denver, which is a mile in the air, lives there for 3 weeks, comes back to San Jose. They have indeed increased their red blood cell count. Are they outperforming? Are they doing better because they have more red blood cells? Well, the athletes think yes. They think if you live in Tahoe, train at higher altitudes, and come down to the valley to perform, you will get, shoot, for them, 2 seconds might even win a race, right? It's a really big deal. Okay, enough of the stories there. Leukopoiesis is the making of white blood cells. There are cytokines, which are, cyto means cell. There are cytokines that are chemicals that signal stem cells to become white blood cells. So I just went really vague here because I told you the name was erythropoietin to become a red blood cell. There are so many different kinds of white blood cells and different names. I'm just going to tell you in general. Cytokines are the names of the chemicals that are used to signal those pluripotent stem cells to become white blood cells. Okay? And the disease here, if you have too many white cells or immature white cells that are out of control growth, that is called, what's that disease called? Leukemia, right? It's a type of cancer where the cytokines perhaps are out of control, making too many white blood cells, and usually they don't mature, and so someone's immune system usually goes down, even though they have all these white blood cells around. So that's called leukemia. All right, blood typing. You probably already know this. Do you know your blood type? What type are you? If you don't know, you should do it in class, in lab. Check your blood type. You are one of 4 blood types. You are A, B, AB, or O. And the antigens that you have that determine what blood type you are the proteins on the plasma membrane of each of your red blood cells. So each red blood cell has a bunch of proteins on the membrane, and they say this is an A cell, or this is a B cell, or it has A and B, or it has none. And the none would mean 0, or O, sorry. Type O would have no antigens. Antibodies are proteins in the blood that recognize foreign antigens. So you also have antibodies in your blood, and they seek and destroy by binding the foreign antigens. Okay? So if I'm A, I don't really like anybody else except other A's. So I'm not going to, if I see a B, I have antibodies that are going to destroy it. Okay? So we have antibodies that attack things that are not similar to ours. If they bind to the foreign red blood cell, then you would get clumping of the red blood cells, and that term is agglutination. Agglutination is the clumping of the red blood cells, and notice it has 2 G's, A-G-G, agglutination. Okay, so let's do a little bit of drawing so I can explain to you how this works. And what we're going to do is draw some pictures, so get a page somewhere, piece of paper, and we're going to do some drawing. So let's start by drawing some cells with antigens. Okay, so let's draw, oops, that's not what I wanted to do. Go back. I need to go down to the bottom here. Sorry, I forgot. I need the felt tip pen. Okay, we're going to draw cells with their antibodies. Antigens. See, I got to catch myself. Why do they make these words so similar? With their antigens. So let's make a cell. 1, 2, 3, 4. So cell A, type A person, let's say they have squares. Antigens. Oh my God, how many times am I going to say that wrong? Antigens say in the shape of squares. Now they're really not, but we'll say that they are. Okay? Type B person is going to have, say, triangles. This might be different than how your book is showing it, but this is how I'm remembering it. So that's how we'll do it. Now, an AB cell, A, B, has both. It has a triangle and a square. Square, triangle, square, triangle. It has both. Both antigens, both A, square, and B, the triangle. Now, type O has none. Alright? Now, as I told you, you also have antigens that notice. Oh my gosh, Lisa. You also have antibodies. Am I insane? Did I not get coffee? Antibodies that notice foreign things. So a type A person likes squares. Do they like triangles? No, they don't like triangles. They've never seen a triangle. They only are going to enjoy seeing other squares. Any triangle that comes by would have to be foreign. So it makes an antibody that kind of looks like it would attach to a triangle and have like a flag sticking out. And we'll go, oh yeah, I've got a bunch of these flag antibodies that are going to attack a B. Okay, so let's draw the antibodies that you would see. Here's the flag. In a type A person, they would attack triangles. Okay? Alright, now a type B person, they like triangles. Do they like squares? No, do not like squares. So theirs are going to be shaped to kind of attack a square shape. And there's a little flag saying, help, help, come get rid of this foreigner. Okay? AB person, do they like squares? Yeah, they have squares. They like squares. Do they like triangles? Yep, they like triangles. They have triangles. They have to like triangles. So what should I draw down here? Nothing. I don't draw anything. So they have no antibodies to A's and B's, because they would be attacking themselves if they did. So it's not safe to have antibodies to A and B, because this is A and B. Type O. Do they like seeing squares? No, they don't have squares. They've never seen a square in their life. Do they like seeing triangles? No, they don't like seeing triangles. They've never seen a triangle before. So what should I draw down here for their antibodies? Both, right. I would draw, they have antibodies to triangles, which are B's, and they have antibodies to A, which are the squares. Okay? So A has A antigens, antibodies, that we call these anti-B antibodies. We call these anti-A antibodies. No antibodies. Anti-B and anti-A antibodies. Okay? So let's see if you can do the question. Okay? Blood transfusions. If a type A person is given type A cells, the A cells will be accepted. Right? A is given A. That sounds good, right? They would be okay. If a type A person is given type O cells, the O cells will be accepted. Well, let's think about that. if you took, where's my pointer now? If you took this one here, okay, the O cells, and you gave it over here to the A person who has anti-B, will they be attacking the O? No, they're only attacking B's, and the O has no B, so it's safe. Okay, next it says if a type A person is given type B cells, the B cells will be destroyed. Well, let's see. If you give this B cell to this person, sure, they're going to attack them, right? They have anti-B antibodies that are going to attack that. And then let's do the last one before we read it. The last one is to give AB to the A person. Are they going to accept them? Well, the squares are acceptable, right? Because we have squares. But the triangles are not. And since they have anti-B antibodies, they're going to attack the AB person. Okay, so it says if a type A person is given type AB cells, then the AB cells will be destroyed. Remember when we say destroyed, we really mean agglutinated, right? We mean that they're going to clump and then the body's going to get rid of them. Alright, so what I want you to do is take a second. This is your group question here. I want you to answer these last two parts here. 1. Which blood types can O accept? And 2. Which blood types can O donate to? Okay, I want you to turn off the show. Take a couple minutes to think about this. Okay, so which blood types can O accept and which blood types can O donate to? So let's go back to the picture again. If you have O and you want to give it to everybody, can you give it to A? Yeah, it's okay because it has no antigen. Can you give it to B? Yeah, it's okay because it has no square antigens. Can you give it to AB? Oh yeah, sure. Look how happy they are. They don't have anything to bother anybody. Can you give O to O? Yeah, you can always give O to O. You can always give one to the other. Now the question is, who can O accept? So here's the antibodies of O down here. So now, can we give O to O? Yes. Can we give AB to O? No way. Double attack. Can we give B to O? Because they have anti-B antibodies. Can we give A to O? Nope. We would attack them too with our anti-A antibodies. Okay? So we often say that since O can be given to everybody, you can give this O, since they are free of antigens, you can give them to everybody. We often say that you are the universal donor, right? Now think about it. Who is the universal acceptor? Who can accept everybody? Now to accept everybody would mean that you're friendly and you'll take them all and you have no antibodies to bother anyone. So who has no antibodies? A, B. Right. So you can give A to AB, B to AB, AB of course to AB, and O to AB. You can give all of them. Sounds good? Okay. Now there's another story. It's called the RH factor. And this was first discovered in the rhesus monkey. It's called the RH factor because it was found in the monkey, and it's just another antigen found on the red blood cell. So instead of calling it, let's see, what would be, we've done A, we've done B, they need a C, right? Instead of calling it C or D, they called it RH. And if you have the RH, we say you're positive. So you would say you're A positive. And the person who has RH, this person has RH antigens and no RH antibodies. So let's draw this too. Where's my pen? So if you are RH, let's say you have RH. You're RH positive. Let's give you circles. Okay, you have circles, circles. If you are RH negative, no circles. Alright? What about their antibodies? Let's draw below here the antibodies that these people have. Does this one like circles? Does this Rh positive like circles? Yeah, they do like circles. So they have no antibodies to circles. That would be bad for them, right? Oops, bodies. How about this person? Do they like circles? No, they don't. So they are going to have, let's see, how can I draw this as like a flag with a circle? It's going to attack circles. And these are called what are these called? Tell me. Anti-RH antibodies. Okay? So, the Rh positive person has antigens, which we call Rh antigens, but no antibodies. The Rh negative person has no Rh antigens, and they therefore have to have antibodies to Rh. Okay? So as it says here, if you have the Rh factor, we say you're positive. The person has Rh antigens and no antibodies. If there's no Rh factor, you say negative, like I'm B negative or O negative. This person has no Rh antigens, but does have Rh antibodies. Okay? So let's do, I needed a blank slide. Let's practice a couple of these things. Okay. Sorry, now you have to see all my mess here. Let's insert a new slide. Remove the background and go back to our show. Well, I just wanted to practice with you a little bit. What would an O positive person look like? What would their cell look like? Okay, what's O positive? What should I draw here? Should I draw squares? Nope. Should I draw triangles? No. Should I draw circles? Yes. Okay, and what kind of antibodies do these people make? They make anti-A, right? They don't like A. And they make anti-B. Do they make anti-RH? No, because there's the RH. They can't do that. Okay, let's do AB negative. What do they have? Do they have squares? Yep. Do they have triangles? Yep. Do they have circles? No. What kind of antibodies do they have? Anti-RH. They don't like circles. Okay, you getting this? We'll do one more. Let's make a B positive. B positive. Do they have squares? No. Triangles? Yes. Circles? Yes. So they are anti what? Anti-A. Are they anti-B? No. Are they anti-RH? No. Alright, so now my question to you is, now that you know about the Rh positive negative thing, which one is the universal donor now? Because before we said it was O. So is it O negative or O positive? And we also did, who's the universal acceptor? And who did we say it was? We said it was AB. Is it AB negative or AB positive? So I want you to stop the show for a second. See if you can work on these. Okay, let's do it now. Who's the universal donor? Why was O the universal donor? Well, we said O had no antigens around it, so nobody was upset. So which one has no antigens around it? O negative or O positive? That's right, O negative. O negative has no antigens, so no one's going to be upset. So the universal donor is O negative. The universal acceptor, remember, was AB, and we said it was universally accepting because it didn't have any antibodies, so it was going to accept everybody. So now which one has no antibodies? AB negative or AB positive? AB, which one? Positive, right. because this has A, has B, it has RH, it has no antibodies. Where's my pen? No antibodies. So therefore, would you like to be O negative or would you like to be AB positive? Well, this brings us to some more stories. The RH factor and the pregnancy of the RH negative mother. So here you have cute little baby coming along. Mom has her first baby. She is RH negative. Okay? So if we were to draw her cell, we would say she is negative. So she has no circles on it. But she does have anti-RH antibodies, right? To attack any circles. So remember it would look like this, however we drew that. Okay? First baby comes out RH positive. She's what? She is negative. Do negatives like positives? No, they don't. So, during the pregnancy, the red blood cells are way too big to cross from the baby to the mother. They don't go through the placenta. They are too large. Only small things like oxygen, glucose, antibodies could cross. Cells are way too big. So the negative mother and all of her antibodies that she has never sees the positive baby until birth. Now at birth, there's tearing of the uterine endometrium and the placenta. And so there's mixture now. There's blood everywhere. If you've seen a birth, it's everywhere. There's now mixture of blood. And so the mother now becomes exposed to RH positive red blood cells. And her body says, oh my, we do not need these in our body because we are a negative. So we are going to make an army of these guys. Okay, so now you have an army of a bunch of anti-RH antibodies. Baby goes home. Mom never sleeps. Everything's fine. And then she decides she's going to do it again. Make her life more crazy. Have another one. Okay? Second baby is RH negative. No problems. Mother is negative. Baby's negative. So although she's got this army, she is not going to attack it. And by the way, the antibodies, the army of antibodies, can cross into the placenta. Antibodies are small. They can cross into the fetal system. But there's nothing, I mean, they will float through and they will go through, but there's nothing there for the mother's antibodies to attack. Problem comes when third baby comes. Third baby is RH positive. Well, unlike the first one, where the mom didn't have very many antibodies, she now has an army of antibodies. And those antibodies will cross the placenta during pregnancy and attack the baby's red blood cells. And this will actually kill the fetus. It's called erythroblastosis fetalis. So a long time ago, before we had a drug to help fix this problem, the government required that people got married and had a blood test to see if the mother was negative and the father was negative, you're okay. If the mother was positive and the father was positive or negative, it was fine. The problem becomes if you have a negative mother and a positive father, because then there's a chance of a positive baby. This is genetics, right? Well, a couple things started to happen. One, people stopped having children after they were married. They were not getting married, not having their blood type, and they were having children outside of wedlock. And these were actually problems that were starting to happen. So the government couldn't really help by asking people to do a blood test if they didn't have to get a marriage license. But in the 80s, there was a lot of research done to help design a drug so that a negative mother, when she has her first baby, she can take, to prevent the army from happening, she can take a drug called Rogam. I think it's spelled like this, Rogam. And Rogam is a drug that will prevent the army from happening. So that when she has a third baby here that's positive, she won't have the army ready to attack. And they give the Rogam, I think, a couple times, not just once, but maybe twice or 3 times. So that after the first birth, they may give it before the first birth, they may get it after. They may do it even though this first baby is negative. they may do it anyway. They may go, oh, you're negative, Mom. We're giving it to you now, just in case for the future. Okay? I do know someone who's involved in this study. It was done at Stanford University to design the ROGAM. She had the erythroblastocyst fetalis like 8 times. She had 2 children. She had 8 miscarriages. She had so many antibodies. They were always using her blood to do all these experiments. Finally, she had, with the ROGAM, she had another baby, and I think it was like, I don't know, 10 years difference between her first 2 children and her 3rd child. So, success story, but it took her a long time to have that 3rd baby. Do you know your blood type? I've asked you this. Look at this is the amount of O positives. In the United States, this is changing as we get people from different countries moving here. O positive is the most popular in the U.S. right now. Negatives are harder to find than positives. Take a note of that. If you donate blood and you're a negative, sometimes you get a phone call saying, hey, we're having a negative drive. Could you come down and donate? Even though O is the universal donor and O negative is the universal, right? We said. You would never give a different kind of blood. You would always give the same. So if I'm O negative, they will find someone at the blood bank who will give me O negative. You never want to say, oh, I'm AB positive, but you can give me O negative. It's never good to do that. Even though it's possible, and we talk about it all the time in class, we would always give the same blood type. And now, today, a lot of people know that they're having surgery. They go donate their own blood. And we had this whole scare of AIDS and all these things. And so a lot of people are very interested in doing that, donating their own blood for a surgery they're having a couple days later. Or someone in their family that they know has the same blood type. So, like we said, in a disaster, you want to be which one? You want to accept from everybody, right? In a disaster, so you want to be AB positive. Look at not very many AB negatives. So we'll have to see what you guys are in class. I'll have you raise your hand and tell me. Fun fact, there is a very rare blood type called AH. Less than a dozen people have this blood type, so you would not want to have that one, right? There'd be no one left to donate to you, except 11 people in the whole world. Okay, our next topic is 3 steps of hemostasis. Okay, we've done the word homeostasis. This is hemo. So what's hemo mean? Hemo means blood. Stasis means stable. Yeah, so we're going to keep your blood stable. So this is how we're going to do it. If you start to bleed, cut your arm, the vessels that are broken or cut will vasoconstrict. So vaso means blood vessel. Constrict means to squeeze shut, right? So you're going to vasoconstrict the blood vessel. And there will be a platelet plug formation. So the platelet will hey, this is how it will happen, okay? There will be collagen, which is in the layers of the blood vessel. There's collagen, which you know from anatomy. It's a protein. Keeps you young looking, okay? The platelets are activated when they come across the fibers of the exposed collagen protein. And when they do that, they make ADP. This is the same ADP as ATP, the same one you know. ADP attracts more platelets. Says, hey, everyone come on over here. And a platelet forms a plug. All those platelets form a plug. They all slap on top of each other. There is something at the time release called prostacyclin to prevent too much sticking. You don't want it to be such a big plug that the blood can't flow. You just want it enough to stop the blood from exiting the hole. Next, there's formation of fibrin. So there are 2 pathways if you look in your book. The first is prothrombin converted to thrombin by factor 10. So that's not an X, that's a 10. Okay, factor 10, Roman numeral 10. To do this, you need vitamin K. Thrombin converts fibrinogen, which we talked about earlier, to fibrin. The fibrin is stabilized with factor 13. And now you have a blood clot. It's good to have a blood clot, but if it floats away, that's called an embolus, which can get stuck in your brain and cause an embolism, which usually means death. If someone says, my so-and-so had an embolism, you go, oh, wow, I'm so sorry. That's usually a death. A clot in a healthy vessel is called a thrombus. Some people get this as they get older, don't get as active. It becomes a problem if someone's not moving. they worry about some old people when they're traveling in a plane because they're sitting so long getting a thrombus. So it's important to move the blood through those lower limbs. There is a drug called Coumadin. Have you ever heard of this one? Coumadin? A lot of people go on Coumadin. Let me write this down for you. Coumadin, I think, is spelled C-O-U-M. Coumadin. The Coumadin blocks vitamin K. So if you block vitamin K, then you don't get this action happening here, because vitamin K is needed to convert thrombin into prothrombin into thrombin. Okay, so Coumadin is a drug. If someone says I'm on a blood thinner, it doesn't really mean that they're making their blood thinner. It's not like erythropoietin. I don't know why they use that word, but what it is, though, is preventing them from clotting. So if someone's on Coumadin, they need to go in often to have blood clotting tests done. If it takes 3 minutes, if they nick them, it takes 3 minutes for the blood to clot, that's way long. It's too long. Internal bleeding could happen and it would never stop. So Coumadin, the goal is to prevent unnecessary clots in the body, and this is something you would be prescribed usually as an older person. But you don't want it too much Coumadin. Like someone who had a heart attack, of course, would be interested in this because you don't want this clotting in the heart, right? But not too much because it could cause internal bleeding. And the other story about this is factor 13. Factor 13 is the one, it's a genetic problem. For those missing factor 13, they are called hemophiliacs. So hemophiliacs are missing factor 13, and they can bleed to death just simply by cutting their foot. Okay? Alright, let's do a little anatomy review. Do you remember any of these words? Oh yeah. Blood leaves the heart through what? Arteries or veins? Through the arteries, right. And so here's the aorta coming off the heart, and then you have the 3 layers, the tunica media, externa, interna. You have elastic layers. Over here, the vein, much larger diameter in the lumen than over here in the arteries. Thin tunica media interna. Here you have this thick muscle layer of the tunica media. Medium-sized artery. Arterioles, of course, just have a little bit of muscle tissue around them, not very much. And then the capillaries are simple with one cell layer. So you have diffusion of nutrients and oxygen and stuff. They're fenestrated, so things can get in and out. Right? The other thing about veins in venules is that they have valves to prevent backflow. So as the fluid goes up back to the heart, you don't want it to come back down again because there's no pump, right? There's only one pump. There's no pump at your feet. So to prevent it from coming back down again, you have valves to prevent backflow. So it's only one way. All right, so here's a picture of the valve. When you move your legs, when you contract your legs, you can actually cause the fluid inside to be squished, and it can't go down because of the valve, so it must go up. So walking, like the example of the airplane, or moving your legs or doing exercises or walking will help that person move the blood up to the heart again because it can't go down. Now if you sit here in the plane, you don't move, you would get like pooling of blood in this area. Well, the pooling of blood can increase the chance for clots in this area. Now you're all young, you don't have to worry about this, but when you're 80 and you decide to go on vacation to Jamaica and can go, yahoo, my last trip, and get excited, you can walk around a little bit and prevent that from happening. And you can see this in class too. If you hang your arm down and look at your hand, find a blood vessel. If you push down like this, you'll get like a bulging right here where the valves are. So I can help you find some valves in your hands during class. The other thing about the blood vessels that we need to review, oh yeah, look at this gross stuff. So as you know, cholesterol gets a bad rap, and it gets a bad rap in this case because there's damage to the blood vessel. Cholesterol sticks, fatty crystals grow, ulcerations, is sick, yucky stuff, right? This is atherosclerosis. So what happens, our question then is, what happens to the flow through this tube? Well, obviously it slows down. Well, if this is feeding your heart, and this eventually cause a heart attack, myocardial infarction. So we want to make sure that we're not getting damage to the vessels. No matter, you could eat thousands of grams of cholesterol a day, and it wouldn't be a problem if you didn't have any damage. The cholesterol is going to get stuck if there's damage. Okay, so damage happens mostly due to high blood pressure. If you have a high blood pressure, you're putting a lot of stress and strain here, and due to age, you're going to get damage, and that's going to make all these things happen. So most of you, cholesterol is not a problem for the younger people because they don't have damage to the blood vessels. But as we get older and there is more damage to our blood vessels, the fat and cholesterol does stick to it and causes a problem. So there's a procedure they can do with, it's called angioplasty. Have you ever heard of this? If this is in your heart, a really small blood vessel in your heart even, they take a tube, they run it through, and they put a little balloon right here in this spot. And then they pump in air to make a balloon enlarge in this space, and it flattens out the crystals here. It smashes them, breaks them up into little pieces so that they float away. Way cool. That's called angioplasty. Okay, other things they can do. One more thing they can do is called a stent. S-T-E-N-T is stent. And it's a piece of metal. Kind of looks like a circular ladder, kind of. It looks like if you took some chicken wire, you know, like on a fence, and you rolled it up, that's kind of what it looks like. And if you stick it into this hole, and it has spring loaded, and you let go of the spring, it goes boing, and it opens up and pushes this flat, and now you have a piece of metal, circular metal, going like a tube up and down from maybe here to here, all the way across. And so it's a tube metal that's spring loaded, so when it opens, then the fluid can flow through, and this has all been smashed down. Pretty incredible stuff that they can do. Cardiologist, anybody want to work in cardiology or work in cardiology? Fascinating. Okay, so to finish up the anatomy review. Arteries, arterioles, capillaries, venules, and veins, you should know that. Veins are distensible, right? They're not as hard and solid as a artery. You can really push on them, and they can go out and they can go in, and more like a balloon. They can go out and they can go in. They're not so hard. They are low pressure. So remember, we're coming back to the heart, and the pressure is really low in the veins. 60% of the blood is in the veins. Only 40% are in the valves, or in the vein. Here we go. 60% are in the veins, 40% are in the arteries. Venous return, that means moving the blood back to the heart, is assisted with valves and the skeletal muscle pump. So the skeletal muscle pump helps push on the vessels, and they have to go up because the valves prevent the blood from going back down. And the diseases, the one that I showed you was atherosclerosis in the picture. The other one is arteriosclerosis. This is hardening of the arteries, and that becomes a problem with blood being pumped, you know, whoosh, 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 as it makes its way out of the heart. If the arteries are not able to take that pressure because they're hardened, then you're going to get potential breakage and damage, and that will lead then to the plaques, and that will stop the flow through the heart, which leads to a heart attack. Okay? Oi, oi, that was a long lecture. We did it. Okay? Thank you.